So today, at the end of the day, Emma will talk about germline copy number, but between now and then, we'll talk about the somatic, the cancer use cases. I gave you an introduction on Monday quickly. I'm going to do a slower version of it today. I'll kind of do an overview of the two pieces. There's two different sets of tools. There's SNVs and Indels from UTech 2, and Aaron will teach you that. We're going to do a 10-minute break after I'm done, and Aaron will teach you about that. And then I believe before or after lunch, I think after lunch, we'll talk to you about copy number and show you that workflow there. Okay. It's very important to understand. So this is a graphic from a paper that is, I find, a little bit hard to understand, but I didn't write the paper, so I don't get to choose. What this is trying to show you is that as a tumor grows and gets older and expands, the pieces of the tumor start to differentiate and diverge. As new mutations start to occur inside of it, some of them become driver mutations and start to cause more problems within the cells as they replicate. So imagine you have one cell that has made changes, it replicates, and now there's eventually 100 copies. If one of those cells has a new major driver mutation, only its children will start to have those mutations, and the other 99 will not. So they diverge. So within a tumor, there are many, many different tumor cells, and all have very, very different signatures, mutational signatures, many different types of variants. And they could look very, very different. So when you if you were going to take a piece of this tumor for sequencing, you'd grab a whole bunch, you'd grab some of the red and the yellow and the greens, and they'd all come together, and that's what you find. So this is very important, unlike in the germline side, where you expect half of the DNA from your mother and half from your father, and we know you have a 50-50 relationship on your heterozygous SNPs. That's no longer true in cancer. Okay. There are many reasons that cancers are caused, and there are many things that cause these driver mutations also. It can anything be from environment, we know, with radiation or smoking or anything like that. Sometimes you have a higher risk for certain types of cancers. So for people who have a BRCA mutation, I don't know if you know Angelina Jolie. She very famously has one of the BRCA mutations, BRCA. And so BRCA is a gene that's very good. It's very important. The BRCA1 and 2 help repair damaged DNA. And so when there's a mutation in BRCA, so BRCA is not bad. You say, I have BRCA. Everyone here has BRCA. Great. It's very important that you do. If you have a copy that is mutated and is bad, you're no longer able to successfully or accurately fix DNA when it's damaged, and that's what causes tumors. I'll talk a little bit about that in a bit. So you can inherit, not inherit the cancer, but you can inherit the higher chance of getting cancers, of creating these mutations. Okay, so there's many different things that can affect both on the normal and the tumor side. This is, I kind of showed you the picture before 2 slides ago. This is a cross section of what a tumor might look like. And look how different it is amongst the tumor. The different cells are. Again, this is just a picture. But notice though, when you were to grab some of your tumor tissue, you're also going to pull some of the non-tumor tissue. It's hard to do otherwise. The cells are all over the place. And so usually you think of a tumor as contaminating your normal. Normal just means non-tumor. But actually, when you look at cancer, the normal contaminates the tumor too. And so it gets very confusing. It's very hard to get a pure tumor cell. On the last day, we'll talk about single cell sequencing, and that actually is a use case very much designed for cancer. But for the most part, cancer sequencing today is bulk sequencing, which means you take a whole lot of cells. So you have a lot of contamination from the normal cell into your tumor. Just like we showed you before, that there's different classes of variants. There's SNPs or SNVs, they're the same thing. Single nucleotide polymorphism or single nucleotide variant. You have indels, you have SNVs, copy number variants, you have structural variants, SVs. Those all exist in the cancer side too. For the most part, the structural variation in cancer are much bigger than in germline. Because again, in a baby, in an egg and a sperm, if you have these big structural mutations, usually the fetus is not viable. Not always, but usually. Okay. In cancer, because it only affects a subset of your cells after the body has already grown, you can have major, major changes. I mentioned this on the first day. I'll say it again. When you think of germline variants, the stuff we've been doing for the last 2 days, well, we know there's noise. We talked about all the filtering we've had to do because there's contamination, or the sequencing isn't perfect, or the human reference is not perfect, or your reads are short. So there is noise in the germline side, but there's a lot more noise when it comes to cancer. We talked about the fact that within a tumor, it's not homogeneous. It's very different. All the cells are different. You have contamination from the normal, from the other tumors. Okay, so everything is different. So the noise to signal, or signal to noise ratio in germline is much better than in cancer. In cancer, you often have more noise than you have signal. And it's also hard because you have different frequencies, or fractions. So usually when you talk about an allele frequency, that's usually in a population. So if 2 people have a mutation out of 100, so your frequency is usually 2 in 100, right, in the population. We talk about allele fraction. That's the percentage of your tumor, of your cells, sorry, that show a particular tumor allele. And that's what's shown on the bottom there. Okay, so imagine I have 3 cells. I just pull 3 cells out and I sequence them. The one in blue there shows 2 normal chromosomes, no mutation, no extra mutations that are somatic, that are cancer. And then I have one I have 4 copies with 1 A mutation. One of them has an A mutation, and one has 4 copies because these are highly mutated, and one has an A and a B. Okay, so we say, so there will be, if you sequence these 3 cells, you PCR them and sequence them, about 20% of the reads will show the A allele, right? Because there's 2 copies of the A in the 10 different chromosomes. And 1 copy will show the B, about 10%. And so that's your allelic fraction. So the A should show up at a 20% fraction and B at 10%. And that's expected. That's normal, not 50-50 anymore. Does that make sense? This is why we don't see a 50-50 ratio in cancer. That's important to understand. Everyone understand that? So when we talk about allelic fraction, that's what that is. Good. Okay. There are so this is why ultimately you'll see the cancer workflow is a little bit more complicated. And when Aaron talks about MUTECT, why the model is more complicated, there are lots of needs for doing things. The number of artifacts just from sequencing is also much larger. So you see this FFPE. I actually forget what it's formalin, formulated something, paraffin. It's basically wax to store, because a tumor is so unstable. It changes a lot, and when you want to pull it out of a tissue and sequence it, you don't want it to move anymore. So they stabilize it by basically freezing it in wax, but it also degrades the tissue. So when they pull it out, the tissue is now a lot worse than if you had just taken blood from your arm. So just storing the tumor degrades it and makes it harder to sequence. So everything about it makes it much harder. And so it means that we have to do a lot of different, we have to think about tumors and cancers differently. Good? Yeah? I told you I'd show you a picture. So this is a karyotyping picture. On the left, you see a normal cell. Is this a male or female? Come on. No one's yelling out. Just yell out. This is an easy one. This is the easiest question I'll ask you. No one's yelling. Just male or female? Female, thank you. Yes, there's 2 X chromosomes. There's no 2X. Okay, good. That was the easiest question. Okay, so notice on the normal cell, everyone has 2 copies of all your chromosomes. By the way, chromosomes are ordered by size. Does anyone know? It's actually, they made a mistake. When they made the human reference, they got 2 chromosomes wrong. You know which ones? 21 and 22. Yeah, 22 is actually bigger than 21. We found out later. Oops, but it was too late then. We couldn't switch them. So, but in general, the chromosomes are numbered based on their size. So 1 is about 10%. Chromosome 1 is 10% of the genome. And Y is tiny, 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 tiny. But now look at the cancer cell line. Look what's happened to, now this is, we've obviously cherry picked an example that's extreme, but look what's happened to your genome. This is a cell, right? Look at the numbers of copies of chromosome 1. There are around 6 copies. But notice the other color. And sometimes you get pieces of other chromosomes mushed over onto chromosome 1. Or some of it is missing. You have whole parts of one of the copies missing. This is an actual cell. This is what happens in a tumor. This is why cancer is really bad. Okay? And I'll just say, just as an aside, this is why chemotherapy, and I don't want to bring everyone down, but all it is injecting poison into your body. Why? Because look how the theory is you'll kill the unstable tumor cells before you kill the normal cells, hopefully, before the person dies. Why? Because look how unstable this cell is. It's crazy, right? You have whole chromosomes missing. You have many copies. You have, again, look at the rainbow over here on chromosome 4. And this is what we're trying to detect when we look for SNPs and indels and copy number. So it's a lot harder. Okay? So this is why you'll have some pity for us when we tell you things, we maybe will make a mistake once or twice. Okay, so I'm going to just give you a high-level overview of the best practices for somatic, and we'll go into the details coming up. Okay, so I'm going to, again, keep it very high, give you the concepts. And the concepts are important. That's why we were repeating them here. Okay, so remember how, so we have, so in the initial thing, we had just haplotype caller before. It was one step. That gets turned into lots of steps. That one variant calling step, haplotype caller, now gets turned into a whole big box. There's a lot that gets done in the somatic mutations. Now, again, we have workflows I'll set up with you. And someone asked yesterday, oh, how can I can't run Whittle on my local cluster here. That's okay. You can see, if you look in, you can see exactly how we run things, and you can just copy it and put it into whatever Bash or whatever scripting language you want. You can run it the same way we do. The Whittle is isn't really human readable, so you can just copy what we do if you want to run it locally. All these tools should be available on the high performance computing cluster too. Okay, so everything, and if not, we'll talk to Terry and make sure it happens. I think Terry's in the other room, so he hears me. Okay. So they're very complicated processes, but we hopefully will make it easy for you today. So first, let's start with SNPs and indels. Okay, so I showed you this. Imagine this is a IGV screenshot. If you can, as much as you can, you always want to have a matched normal. Again, a normal just means non-tumor tissue from the same participant if you're studying it, because you don't care about the germline mutations when you study cancer. They've been in every single cell of the body. They're not interesting. I only care about the mutations that are specific to my tumor, that are driving my tumor. So you want to remove them. And if you don't have the normal tissue, it's very hard to distinguish between what's a somatic mutation and what's a germline mutation. There are ways of doing it, which we'll teach you. I think Aaron will teach you how to do that. But as much as you can, you get a matched normal. And that's what it does. It helps remove the germline variants, because you can see them in both. Oh, I told you I was going to do that. Okay, so you could do tumor only. Sometimes you just don't have a normal tissue. Sometimes you weren't able to get it. It is possible. Aaron will describe to you exactly how to do it. It requires what we call a PON. We'll talk about the PON in a second. A PON, P-O-N, is a panel of normals. Very important, the panel of normals. And the panel of normals generally, in this case, you will use it to also remove germline mutations. But usually the way to think of it is a matched normal removes germline, and a PON removes artifacts. A panel of normals removes artifacts. Okay, so let me explain what that is. We know that there are positions in the genome that are systematically bad. We can see them when we do our filtering. We have a call set of 100 samples, and we filter some sites out because we can see them bad in all our samples. We told you that. Well, guess what? When you sequence a tumor, they'll also be bad in the tumor too. They're going to show up there. And because they are not great, they'll look like somatic mutations. Because we said the somatic mutations look bad. So the artifacts from the sequencing will also look like somatic mutations. So the way to do it is to take a whole bunch of non-tumors from different patients, different people, from anything and see if you see those same artifacts. And if you see them in other non-cancer samples, you have a good sense that these are just artifacts and not somatic mutations. And that's what a panel of normals is. Okay? And they're very important. If you don't have a normal, that's okay. If you don't have a panel of normals, that's bad, because you'll think everything is somatic and real. And we'll teach you how to do that. The most important thing, and again, Aaron will talk about this, I will talk about this, we'll say it over and over again, is to create a panel of normals that's as close to the thing you're sequencing as possible. So if you're running on a NovaSeq machine, you have a bunch of samples sequenced on a NovaSeq. If you're running with this capture kit, use the same capture kit. If you have two different capture kits, create two different panels of normals. If you're running on BGI, have a different panel of normals for BGI. Okay? You want it to be as consistent as possible so you know that the errors are the same errors. And I'm going to keep saying this over and over today. If that's the one thing we come out of today, that'll be a big success. All right. CNVs are also very hard. Almost always in the cancer world, they for sure do whole genome sequencing, but for the most part, I have seen exome sequencing for the cancers. Because again, the mutations are so big, you don't need the whole genome. They're clear. It's happening even with the exome. And it's the genes that are being affected, so that's what they're studying. So mostly we have exome sequencing. But we told you earlier, and you'll see it later on when I talk this afternoon, the exons, as they get captured at different efficiencies. So some exons capture well and get sequenced well, and some exons don't capture well. Some sequencing centers use extra baits to help certain exons, and some don't. And it's very inconsistent across different kits. And different chromosomes are different, and different regions of the genome are different. And so it's very inconsistent. So there's a lot of noise trying to figure out the CNVs. And so most of CNV detection, culling, is just removing noise and normalizing things. Just math to remove noise. And you'll see all about that with me this afternoon. Here's an example. So the way to kind of view this, and I'll show you again this afternoon, is flip it over on its side. So on this left panel, here is the beginning of the genome, and this is the end of the genome. So we couldn't fit it all the other way, so I turned it to its side. And each one of the dots is the average depth of one exon in the genome. So each one dot, for every exon, there's one dot. And it goes across, it's plotted here on the y-axis by its position in the genome. So chromosome 1, and this is chromosome X here. And here, the further out to the right, the higher the depth. Look how inconsistent you are there. Where are the copy? So if you only got to look at this left plot, where are the copy numbers? It's very hard to tell. So what we do is, again, a lot of math and stats to normalize the coverage and pull out. And we'll teach it to you all this afternoon. And again, same thing here. I'll say again, the panel of normals is very important, just like it was for SNVs and Indels. You want a panel of normals to tell you, hey, look, I know that this particular exon happens to capture very well. And it's always at high coverage in every one of my samples. So when I see it in my tumor, it's not a copy number event. It's not an insertion. Or this exon captures poorly in all my samples. So when I see no coverage in my tumor, that doesn't necessarily mean it's a deletion. And that's what the panel of normals helps you do. It helps you normalize. So it's important for this also. Okay, that's the overview for the somatic side. I will take questions. You're going to learn it all in detail. We're going to jump into detail. So we have a 10-minute break coming up, I believe. And then Aaron, I think, is up to teach you all about MUTEC. I think that's how the day would go. Give me 1 second. That's a yes. I have agenda here. All right. So we're going to take a 10-minute break, and then we'll learn about MUTEC 2, and you'll get to play with it hands-on. Then after lunch, we'll do copy number, both somatic with me and germline with Emma. Good. All right. Any questions for now in this room, and anyone in the next room, please come by. Good. All right. See you all at 10:55. Anyhow,
Hi, I'm Aaron again. I'm going to go into a few more details a bit on the MUTEC-2 calling pipeline. Okay, we talked about the concepts at a high level. I'll go into a little more depth on some of the concepts here. And in particular, I'll go through, like I said, exactly what does that pipeline look like a bit better. So at a high level, here it is, right? And it looks complicated, but really, it really is not. And it's not that different than the standard calling for just germline stuff. At first, you got a pre-processing stage, as take a BAMs, align them, try to have a basic analysis ready BAMs. And then in the middle, you have your variant calling stage. That stage has a few more parameters here. For instance, MUTEC-2 takes that part right there, by the way, is analogous to when we just used a haplotype caller before. That middle area, think of that. In fact, Mutex's built on top of the haplotype caller, all right? So in that middle stage, you have a few extra arguments though. As mentioned before, it takes in the TN pair. That's actually, it's a paired file that doesn't just have your reads for your tumor cells, but also reads from normal cells from the same sample, okay? Again, how is the problem space different for trying to call somatic variants is that you only want to call the ones that aren't in the germline. And so we pass in the paired file here. Further, we also have the panel of normals. I'll talk about that in more detail later. It's very important. That is technically an optional argument here, though. Use one if you can, but I'll go into more detail on that later, okay? There's also the germline population frequencies. Again, more information that helps us try to make the call, not just am I sure that this is a real signal I'm seeing in the band files? Is it a real variant to call? But further, is that variant a somatic one or a germline one? So we can use frequency distributions of just sort of normal variants in order to help tell that. All these things we use them. I'll go into detail a little more about how we use them, okay, to up our confidence. And also, one thing I should mention though, side note, some users find their way to MUTEC-2 because they're trying to discover one particular type of event, the LOH event, loss of heterozygosity, where you have a het call and then in the somatic cell, you lose one of them. And now it looks like a HAM call. MUTEC-2 is actually not the best tool for that, okay? We actually have a separate one called allelic CNV. Okay, that's a specific tool for that. This one it might sound like it's good for that, but we have a specialized tool just for detecting that case. Okay, so those are the particular types of events you're looking for. This won't really be the best tool for you. All right, but to move on, like I said, I'll go into more details on what the arguments are and why we use them in the variant calling section. But then we have filtering. Definitely go into filtering. There are some metrics that come out from the equivalent of the haplotype caller in MUTEC-2. There are some metrics that come out, and when we use them as well. There's a hard filtering phase. There's another more sophisticated model that we also use to filter. I'll talk about that at the end as well. So there's a lot of extra filtering phases here. And then at that point, we're pretty confident. After variants, you can go and you can annotate them using the funcatator, and then you take them off and it's analysis ready. You're fairly confident in the variants. You annotate the ones that are there, and you can move forward. So when I mention false positives, I bring it up a lot because that's really false positives are even in our germline cells. A lot of our filtering is all about reducing, false positives. It's a little more important here when you're dealing with potential tumor cells, right? We have a lot of extra steps in here to really try to bring down the false positive rate. And to do so, we should understand where false positives come from. Now, some of the false positives are coming from the exact same places they would in a germline cell, right? Like over here on the tumor cell here, you see a possible evidence for a variant there, possible evidence, right? But there's some strand bias present. And like you can see, almost all the evidence is coming from just left reads and not right reads. That strand bias is still there. All the standard problems we have in calling are still there, and they don't go away just because they're inside of a somatic cell, right? So this example of we still have the standard issues. There are still areas that are hard to align to. There are still going to be artifacts that we have in reading. Some even worse. But the thing is, there are also other classes of issues. Because on the right, you start looking at something unique to this problem space compared to germline calling. What about here? You have very good evidence here for that you have what appears to be a variant here, right? You don't have enough evidence. A little bit there. You can see it in the germline sample, but the problem is the germline sample there has low coverage. So you see some evidence of it, but maybe not enough to call the variant. Doesn't mean it's not there. It just means that you have a difference in coverage, right? But the goal is to really distinguish which mutations are unique to your tumor cells versus the germline one. So that's another source of false positives. You might call that as being, hey, this is novel to my tumor, and yet it's not. So there are some tools we have to also try to get rid of that case as well. All right, and in general, we have, our filtering and also our panel of normals, very important. We will go into that in more detail. We try to use them to get rid of just the kind of basic sequencing artifacts we have, right, that are everywhere. We also use things like population distributions, like Nomad, to try to determine when you might have a case like this, where you have some evidence for a variant down there, but you don't know whether it's enough. We can pull on other population information, germline population, and say, well, maybe that's a very common mutation there. So maybe even though it's not enough to call it, together with population information, we can say, we don't think that's the unique mutation in the somatic cell. All right. And by the way, that happens sometimes if there's limited funding and people want to focus their sequencing, they'll focus a lot of it on getting really high depth on the somatic cells and maybe a relatively low pass. But it does introduce this class of error where you don't have enough depth to be able to call it in the normal cells, but you can on the somatic cells. But then you also can make the mistake of assuming it's only there in the somatic cells if you're not careful. So we talk about artifacts, but how do we get rid of them? Well, BQSR, we spoke about that already, right? Some parts of it will look exactly like our normal call line. Also, our panel of normals really help figuring out artifacts. I will go into a lot more detail on those as well, but the idea that you ideally you run samples unrelated to the actual person from which you're taking the somatic cell, the tumor cells, and the normal. These are unrelated people run through your same pipeline. They also help us to discover what might be artifacts of your sequencing. And like I mentioned, we also use variant site databases. dbSNP and EXACT are great for the germline side, and COSMIC is good for somatic. So like I said, what if you don't have evidence? You don't have enough evidence to say this thing was probably a germline mutation. Well, what if you find that that mutation is present in dbSNP or Nomad and is very common and is not really referenced in a specific somatic database? Well, that gives you more evidence, or vice versa. You see, well, actually, that's not a known common mutation in the germline, but it is present in a lot of cancer cells. Again, that's evidence the other way. So using these kind of distributions can really help making the calls in edge cases. And things can get ambiguous a little bit when you're talking about somatic cells for many reasons we'll go into. And I'll point out that we're doing something a little more sophisticated here in GATK4. Okay, we're not just going to go in and say, well, I have a list of variants called in the normal tissue, and I'm just going to subtract them away from everything I see. As we mentioned, just doing that will actually give us more false positives. There might be evidence for it, but not enough. So we're actually putting in we're feeding in and analyzing the raw BAMs from both. It's not a simple subtraction. We're combining various sources of information to get more statistical confidence in our calls. But it's a little harder than just saying, hey, I see these calls in my normal sample. I'll kind of sequence them separately and I'll just subtract them off. We got to do a little more than that. And I'll go into a little more detail about that too for you. So again, that panel of normals, we mentioned a bunch of times. So normals, we mean that they're not cancer cells. We're looking at basic germline. We're assuming that the people that we're including in the panel of normals we're not like finding tumors in them. They're just normal germline cells, okay? In a perfect world, what we do is we run them through the same data generation techniques. Okay, if you're using Illumina sequencing, run them through. In a perfect world, you want to do this, okay? And again, if you do exome capture, well, try to use the same exome capture kit to eliminate variables here. And the idea is, as much as possible, let's say you have a very, low frequency mutation somewhere, right? It's low frequency, but weirdly, you see it in all your panel of normals, and you see it in your somatic cell. Well, that's evidence Not conclusive, but that's evidence that maybe the thing you're seeing is actually an artifact of your sequencing. Because it's unlikely that a series of randomly chosen individuals are also going to just so happen to have a mutation at that same spot where you see it in your somatic cells. You see? So having unrelated people go through your pipeline, that's one of the ways we can identify what might be artifacts from just the process you're using. Remember, we're really focusing very heavily in this case of reducing our false positives. So any additional information like that is helpful. And I want to point out that even if you can't technically, even if, let's say you don't have a paired normal file to go with it, you only have the tumor samples, you can run it that way, but you can run it without a paired normal. You can also run it without a panel of normals. But I really recommend not running without a panel of normals. Even if you don't have a paired normal file, hypothetically, a panel of normals and also population information from from Nomad, for instance, can really help us make confident calls. That panel of normal doesn't just increase our accuracy by twofold, threefold. We're talking several hundredfold increases in our ability to confidently call variants. Okay, even if you have a panel of normals, like on our workflows that we provide on the Broad, you see there's a panel of normals we have there. Even grabbing that is better than no panel of normals if you can. Okay? So we got our actual tumor normal pairs, our panel of normals, our population frequencies. We go into MUTEC, we call some variants. What now? Well, from that point, we move on to our filtering steps. We have several of those. There's a nice little box that says filter variants. It's a tiny box. It does a lot, but we'll talk about it in detail. But let's go into a little more detail in what's happening inside of MUTEC 2. So I mentioned MUTEC 2 is based fundamentally on the haplotype caller. It's doing something very similar. It does assembly around the area of all the different reads, and it goes into call. But there are things we do differently here. First of all, it's going to run on both the somatic cells, and it's also going to run on the paired normal, the paired normal sample you put in, right? And so one thing Eric mentioned, it's hard calling variants on somatic cells. It's hard. One reason is it's not just because there can be obviously weird things like mutations going on there, but you have a lot of potentially different cell lines that are there. Not only do you have different cell lines, but you also have like some normal cells mixed in. You know, your tumor sample it's more difficult, and because of that, that's why we make modifications to the haplotype caller here. So one of the big ones is that one I mentioned here is where we use the panel of normal. This is where we for instance we'll toss out variants. We see them frequently inside the panel of normals. We'll just toss them here. Okay, we'll assume they're artifacts. There are also some that have like, again, if you see a large number of variation in your normal samples, assume that might be a noisy spot. Okay, assume it might be noisy, toss it there too. But when we toss things first and we're getting down to actually calling them, we have a different modes of operation when we're processing tumor cell, what we believe to be the tumor samples versus the germline ones. We actually have a very high statistical threshold for what we'll use to call a variant on the somatic side. For instance, I mean, the filtering is very strict. The mapping quality has got to be greater than or equal to 20. That's a higher threshold we have. If there are two overlapping reads that might look they might disagree on what it is there, well, we toss them both. Again, we're aggressive in tossing information there because honestly, they're like Eric mentioned, even the act of storing the cells sometimes has a tendency to degrade the DNA. Okay, so we want to be really sure we have a high threshold for calling anything on that side. And the flip side of that is when we're trying to assemble potential haplotypes from the normal cells, we're actually pretty permissive, and maybe more permissive than normal. So for instance, any mapping quality. We don't do the Part 20 cutoff. We're looking at normal cells. Well, we accept lots of mapping qualities there, okay? And we'll actually keep, I mentioned potential disagreeing reads that are overlapping. We'll keep them. We'll factor them in. Again, the idea that we want to be very sure when looking at on the somatic side because of the noise that what we're seeing is real. But on the germline side, sometimes you have low coverage. So there are things that complicate the quality of it there. We're fairly permissive. If we see something over there in combination with our background frequencies, we're more likely to give it weight here. It's different than the normal haplotype caller, okay? And the idea is, again, minimizing false positives. And there are also complications. There are changes to the haplotype caller because you might remember in the haplotype caller, after we assemble potential genotypes there, we then go and we compute the likelihood, like is it AA, is it AT, is it TT? We compute those. But things get complicated on the cancer side because because you have different cell populations in there and everything else, we don't always see the reasonable allele frequency there. Like we don't see, for instance, oh, it's a het call. That means it must be 50-50. Well, we might not see 50-50. We might not see that. So at the part where we actually list out our frequencies and calculate likelihoods, we can't use the standard assumptions of ploidy here. That's another tweak we have to make deep inside of it. And I don't worry too much about it, but that's one of the things we're doing here. Okay? I guess any questions there? The takeaway is that we're very strict in saying we think we see a variant in a tumor cell, but actually very permissive if we think we see it in the germline cells. And the idea is we don't want to accidentally say, oh yeah, that's definitely a tumor variant, when it's really not. So we can look at, for instance, here examples of how MuTech2 works versus older technology. So in this case, you got a 128 base deletion there. But the problem is you can see that something is going on, right? A bunch of misaligned reads around here. Something is happening there, but it's really hard to actually call it. Now, a standard pilot-based caller, like the predecessor to the haplotype caller, actually, we get confused there. You might see evidence that there's something there, but you won't find it. And if the actual read lengths are roughly the same, you won't even see evidence there. So they're like discordant read pair callers. Even they can't always work if the actual length of your reads are pretty constant. This is an example where it's really hard to find out what's happening on other technologies like BWA, which I mean some of you who work in somatic areas might know it. But when you run through it with MuTech2, remember it's using built on top of a haplotype caller, which explicitly goes in and tries to reassemble what's happening there with its reads. And because of that, here we actually see, hey, turns out that's a 120 base deletion here. We can see that now, whereas before for older ones, it was harder to see. So that there are cases like this when you're analyzing cancer cells where by building on top of haplotype caller, we can get a lot better information about what's going on, including weird deletions like that, like I said, there's signal there in as much as it being noisy if you're looking at the actual reads in IGV, but here we can go beyond just saying something's happening to identifying, no, we know what that is now. It was a 120 base deletion. So, okay. We've gotten out of MuTech2. We've used our panel of normals. We've used background frequencies. We've made some calls. We're pretty confident in our calls. And now we move on. We're doing, at this point, even more filtering past that stage. Like I said, we want to be very careful in the somatic case. There's further filtering here. That box I mentioned was little, but there are actually several types of filtering that we're doing here that I'll go into in turn, okay? Well, as we mentioned, in many ways, the first test, and I have them listed like this. I'll talk about them one at a time. That's mostly because I can't carry two conversations on at once. I speak quickly, I know, but I do have limits. So first we'll talk about MuTech2, we'll talk about hard filtering, and we'll talk about the model-based filtering here in turn. But to some degree, they're happening together. So again, in MuTech2, we actually filter there based upon, as we mentioned, population frequency, panel of normals, a normal sample. There's a lot of filtering that actually happens there, all right? As we move on to hard filtering, well, we mentioned hard filtering. You just did some hard filtering in your exercise. You can set, we set here too in our pipeline, reasonable thresholds and say, okay, whatever was caught there, whatever we have with confidence, let's do a level of just hard filter it out here, okay? Let's pass it through once. Let's just toss things that we don't think we're confident enough in. Remember, we're, in this case in particular, even more than basic germline calling, we're really trying to minimize those false positives here. So even after we come out of our modified haplotype caller in MuTech2, then we do another phase of hard filtering that would further whittle down. Like if you start with 100,000 potential variants there, we might get down to 50,000 at that point, just hard filtering. And what are we filtering on? Well, okay, well, you can do thresholds and cutoffs as you saw in the last example there. And here, we have a bunch of them that are possible for you to do your thresholds on. We have this is all up on our website. You can take a look yourself at what every one of these are, tweak it if you need to. And I will point out that actually a lot of these stages here have a surprising amount of sliders and knobs to tweak, because some cases are different. Some cases are specialized. We don't necessarily assume we know all the settings we'll need, how much to weigh one thing or another. We give good defaults, but we do give descriptions of all the potential fields that we do hard filtering based upon. All right, you can look this up on our site here. And as we mentioned, even if the header lists a filter, it doesn't necessarily mean that we actually we ran it and we removed things based on it. But as it goes through, we will actually have we'll say whatever filters we are running in there in the header of the VCF file. Okay. And yeah, the filters do require certain annotations. For the most part, they're acting on our annotation. But if you're coming out with data that might not have them, and the filtering step fails, it's probably because you might not have the annotation they need. I do recommend you look up the documentation on these. We do document them, including what annotation that they expect to be there in order to function. Okay. So okay, we've come out of MUTEC 2, our modified haplotype caller. We've done a passive hard filtering. And now in this potential examples, we've cut our variance in half. Now we'll do our final step, which is what we call a model-based filtering. So this is actually a machine learning stage. You have your raw variants, you filter them down, and then you go into a refinement stage. This is a loop. Some of you, those computer science among you, might recognize from optimization problems. You start with a set of assumptions. You say, okay, here's the variance I assigned. Here's the filtering I assigned to them. What is the likelihood? We use actually on the statistics side, we use the F-score, in case those of you in statistics want to know. And we say, okay, what is the F-score? Basically, how likely does our assignment of what is good and bad fit the entire call set? And then, as is the case for optimization problems, we do some tweaking. We say, well, what if actually we change our threshold a little here, filtering? What if that was an error or not? And we say, does that bring the likelihood up for the whole call set? Looking at it as a whole, like, is that a better interpretation of that data to maybe filter that one or not filter that one? And if so, we loop. We go back in, we say, that made it a little better. Let me try it again. Let me recalculate the probability. Let me recalculate which ones are good, which ones are not, and make another tweak. Still, what if I tweak my cutoff for this annotation? Does it make the call set better or worse? Recalculate, back in a loop. What if I make another tweak over and over, using different aspects of it until we finally have confidence that there are no obvious changes we can make that'll make the overall confidence in the call set higher. This is the last step of refinement that will help us figure out what other bits of it do we have to cut away so we are confident in the whole. It will also cut down the set of variants that we actually have left, though. But that's okay. We'd rather undercall than overcall in this particular case. Any questions, though? I'm describing a general sort of, this is how a lot of optimization loops work in algorithms, but I know that might be something, I don't know how many of you have a computer science background or anything, but this is that loop where you go over and you change a model, you say, is it better? You change it again, is it better? Is it worse? And eventually you stabilize and say, there are no more obvious changes to make it better. That last machine learning step refinement there is how we really, really zero in and gain a lot of confidence in our total set of calls there. I will pause though. Are there any questions? How many samples for the panel of normal? I did have this question actually, but before the talk as well. That's a good question. I don't know what the standard would be. My actual area of more expertise is in germline calling. I don't know, Eric. Do you have a good sense of what would a baseline number be for your panel of normals? Okay, yeah.
Overall answer is the more the better. Samples probably the minimum. For the SNVs, I will put the number of 5.
Gotcha. So again, I'll repeat, the question we had over here was, how many samples should you have in your panel of normals? Eric's response was, in some ways, the more the better. 20 might be a minimum, but if you can get better, that's important. What's more important than the absolute number, though, is consistency. If you can match the particular sequencing technology, like, again, if you're doing exome capture, the particular kit you use there, as much as you can match that, you'll do a better job. It will allow it to better identify artifacts and filter them out. Okay, so again, a panel of normals is much better than no panel of normals. All right, but like, as better you can match it, though, the better the results will be. And as he mentioned, there's also slightly different advice on copy number variants, but when we go over that later, he'll mention that particular use case. All right, another question? So can we use RNA-seq data for actually figuring out the allelic imbalance? Okay, that's interesting. I haven't actually used it that way, but Eric's answer that it should work with RNA. Huh, that's a cool use case. Sure thing. Which one was that, Eric? Here, was it this one? All right. Yes. Mm-hmm. Yeah. I see. So at this point, you're mentioning like a different allelic frequencies due to expression because you might have a change in promoter. I believe here we're talking about different allelic frequencies present just in the actual DNA here.
That's how it expressed in shows up in the RNA.
Yes, okay. But you mentioned like a promoter might change, for instance, and you might have a higher expression there at the level of what's being expressed versus it simply being different in the germline. But let.
Are you doing the mitochondria
I am
tomorrow?
tomorrow.
Yeah. So Aaron will do mitochondria tomorrow. What tool do you think we use for mitochondria? Even though that's germline, it's not diploid. So we actually use mutet2 for mitochondria. So I hope I didn't ruin your surprise for tomorrow. But
I mean,
the
a
point is,
little.
we use mutet2 for many different things than just cancer. It's really when it's not when you don't know the diploidy in advance.
I'm just a little ruined. I don't know. What's up? It's based on what then? Okay. Okay. Okay, so the question was when I mentioned haplotype caller behind the scenes, and we use it based upon a certain assumption that what that genome might look like, and does that cause issues when doing with cancer cells here? We make other tweaks to haplotype caller here. There are a series of tweaks. One is, of course, I mentioned we have different thresholds of that we apply to germline. We also the part when we actually compute our likelihoods and call variance based on that, we do change that part as well. I know functionally what they do there. I have not gone into the code and that part of it there, but that's part of the reason I believe why we try to group by allele frequency as opposed to just making assumptions of ploidy and what that would look like. So the fact that we might have a lot of different alleles that might show up at different rates in our cancer cells, we had to change that part so it didn't break haplotype caller. So we can account for the fact that alleles might show up at different frequencies that might not be 50-50 or something else. We didn't want to assume. Also, remember Eric mentioned we might have a lot of different copies there. So that's why we had to change that part. I'm not sure if that 100% answered your question, but that change was meant to account for the fact that alleles show up at different rates. Sorry. If that didn't answer your question, though, do free to come up afterwards. Sorry, you have a question? That's a really good question. Do we have to separate by gender? I mean, the main difference we'd see is, of course, a Y, right? There are different copies of the X. I don't know though.
For the copy numbers, you'll see you have to separate.
Okay. So for some, yes. for copy number, yes. Oh, yeah, so sorry, yes. The question we had up here was a very good one. In choosing our panel of normals, do we have to separate by gender? Like if you have a male sample you're looking for somatic cells and germline cells, would you only want male samples in the panel of normals? And what Eric said was that when you're dealing with copy number variants, actually you do want to do that. But here we don't bother at the level of mutet2 when we're dealing simply with somatic cells. Obviously, there are places where you would see the difference potentially on the X and Y, but beyond that, it should be okay. You can look at you can take them from however you can get them here. Hello, another question. What was that? So 1 second. I can walk away. It's hard to hear from back here. Eric, do you hear the question? Sorry. I'm sorry. After here? Yeah. I mean, after we come out of this stage, we certainly do more filtering here. Okay. Like again, this is mostly to get you your calls, and we also annotate the calls after this. But like we have again several stages of filtering we do here. But I believe we could run if we want various bits of filtering. And actually, if I skip ahead just a little bit here, one thing we do mention is that I mentioned a little bit on the model-based filters here just for a second, and hey, those are the parameters we tweak, by the way, when I mentioned the last optimization stage. You can look them up and document them. And again, annotations here for both of the filtering, the hard filtering and the model-based filtering. We do document all of them, okay? So you don't have to know what they are in advance. You can read up and find the requirements. But I'll mention that it does mention here you can joint call multiple samples. Okay, so in that case like that, you actually could do another level of filtering afterwards, okay? Because you can, like, from the same tumor, you can keep calling them over time until you get changes and stuff like that, okay? So you could do more filtering later beyond the initial stages of heavy filtering we do here. This is actually the last slide. There's more in here. You see that go into more details on certain aspects. Like we talk about, for instance, how we change our modeling to deal with allele fractions. This part I'm not going to have in the actual presentation proper, but they are there in your slides, okay? Calculating contamination. There are details here for how you can run that, okay? I'm not going to do them in the actual live slide now, but they're here. They're here in your slides to take with you, all right? So any more questions? And also mentioned here that, yeah, you can run it on mitochondria. I'll talk a bit about mitochondria tomorrow morning. There's some very, very good set of questions so far on this. Any more? Okay, then thank you. Let's see, who's up next, actually? Anton's up next. All right, then I will turn it over to Anton. Thanks.